one is conducted according to procedures and that it's fair. And so it's received an external person. Leto is a renowned atmospheric scientist, especially with global climate change, CO2 impacts, all that stuff. And I think not much about plant pathology.
Nothing about that. But I think that's the goal.
Why are you
So are you familiar with the sequence of things?
I
think so.
That's not good.
Questions on that. If that's okay, we'll start with the
Joo, hei, Karisto, Jaakko.
questions basically.
Joo, niin mä kuulin tämmöset Joo. Tämä on Petteri Karisto. Ja minä olen
I'm sure personally, I'm a
päinkätään kolmessa suuntaan. Petteri äiti on Joo, tehdään muuta. Miten? Onko sä ollut täyttä?
Supervisors, but usually the supervisors have so many questions over the last years that
käydään vähän omalla on teiltä professionaalia kokeilla. Vähän vuorokäytössä. Joo, okei. Joo. On vähän reisipolta.
Joo, sitä löytyy. Yeah, no, it's very like they were driving us. Yeah, for that for sure.
very
Except for when the actual thing isn't that sort of
Yes, it's
more formal.
Yeah.
Are you?
Unless they're about to take a base.
No, like that.
No, actually, that's a farmer.
It might be.
Well, I think it's buildings that are very yeah.
I don't know.
I guess she was very stressed out. Because I like even more beyond yeah. Good, how are you doing? Up here.
Upstairs.
I'm still not. Oh, down here. Up and down. Is it like
So you can charge
yeah. set this like for yes, like COVID, and I got a little bit, got up it, got a little bit. It pretty frustrates me. Sometimes I'm like,
my first thought when I got there was like, okay, I should get a bike immediately, but it's not.
great. Yesterday I very nearly got it. And I very nearly was quite a serious trip. It's a nice way to go along the mountains, but also went far the most water. It's quite more and how they keep changing the light. And so I thought I was going to bike path. I was coming down the hill really fast, and I thought I was going to on to be still a bike path. But I was kind of car. I didn't see it. So I just went onto the road and parked it. Drink. I jumped on it.
Yeah, they're not super they don't have
yeah, especially not for the cyclists. Yeah, like way.
I really want to go for
yeah, I did. Told me about this. I'm trying to optimize that trade-off, but I still have to send my stuff. Yes,
so you start well, it didn't look so bad.
Can I spoil? What can I say? Mine are still fine, which is impressive.
Much less fun.
That's from BT.
Is there anything else?
I don't think so.
Close the door.
Okay, so good morning and welcome to the PhD defense of Petteri Karisto from the Department of Environmental System Science here at ETH in Zurich. It's a pleasure to see so many faces, and it's of course a great and important day for you. My name is Doctoral. I will be chairing this defense. I have nothing to do with this work. I know nothing about plant pathology. I'm an atmospheric scientist, and the only thing I'm doing is making sure that this follows the procedures as usual. I would like to welcome and introduce the committee, in particular the Professor Emerson Del Ponte from the Universidade Federal de Viçosa. Did I spell this correctly? Thank you very much for traveling this far and for taking the time to examine this PhD. This is an important task. We do rely on outside independent expertise to make sure that the quality of this work is adequate. So you get the most time for questions as a reward for traveling this far. Then Alexei Petteri, if I get this right, also from our department here, who supervised the PhD, and Bruce McDonald, who is the doctoral father of this defense. The way this works is we'll have a presentation from you, 20 minutes or so, followed by questions from the committee and questions from the public if there are any. So the floor is yours.
Thank you. So good morning everyone, and welcome here. It's really wonderful to be here and see your familiar faces in the audience. I also like to welcome Emerson very much here. It's really nice to get you here, and also welcome our audience through the live stream, so you are online. I would like to start my presentation with acknowledgements. First of all, I want to thank my supervisors, Petteri Karisto and Bruce McDonald. I've said lots of positive things about you. I have got a lot of support from you for myself and my ideas, and freedom to follow my ideas. It's been great. I also want to thank all the collaborators. In the first chapter, we collaborate with Andy, Kang, Jonas, and Akim from ETH's Crop Science, and Fabio from Macroscope. In the second chapter, we had wonderful master student Susanne Dora in our group. In the third chapter, Frederick and Sandrine from Indra Biocell, close to Paris. And in the fourth chapter, we collaborate with Sarah, Moes, Hayer, and Amor from Tunisia. I also want to thank all the plant pathology people and also my lovely family for all the mental and practical support during my doctoral studies. The main topic of my doctoral work was Cetoriatritis blotch. It's a disease that is most important on wheat in Europe. And wheat, in turn, is the most important crop in Europe. The annual production value is 33 billion. But if you look at this pile, you might notice that something is missing from the top. The farmers use an extra billion every year for fungicides that they are spraying on the field. And despite that extensive effort, the disease is still causing losses of about 2 billion euros every year. So this is how the pile could look if SDB was not around. But that's not the case. We are losing the money. Now, how does the disease make this economic damage? SDB, Cetoriatritis blotch, is a fungal disease. It's caused by fungus, Cetoriatritis, and it's visible in the field in these brownish or yellowish lesions that appear on wheat leaves. And by damaging the leaf tissue, it reduces the yield production of the plants. How the disease works? There are spores that land on a wheat leaf. On a wheat leaf, they germinate, they grow hyphae, and then that hyphal string finds a stomata where it can penetrate inside the leaf. Then it grows inside the leaf and causes this symptomatic disease after about 10 days. If you look here, you see fresh wheat leaf tissue collapsing and turning yellowish and brownish in this time-lapse video. So this is how the disease looks in the little bit faster mode. You also notice these dark spots. Those are called pycnidia. They are asexual fruiting bodies of the fungus, so they produce clonal spores. And those clonal spores will then splash around with rain and cause further disease. If you look more carefully to young lesion, you see some young pycnidia, these little green bubbles. And if you look them, you see how they start oozing. They push out transparent or crystalline masses of spores. And those spore masses will cause the further disease. You see the spores are coming out like that. So this is how the disease looks in small scale. In more broad scale, generally, we can describe the disease dynamics with epidemic cycle. Epidemic cycle gives the seasonal dynamics of the disease. From the top you see it starts with initial infection in young seedlings. With winter wheat in Switzerland, it would be around October, November at earliest. Then there is lesion formation, as you saw. there are pycnidia produced within the lesions, then these clonal range/dispersed spores, and there can be multiple cycles of clonal reproduction and infection during the season, and that will lead to the devastating epidemics that you see in the figure in the bottom. Later in the season, there is also sexual reproduction. Fungal strains mate and produce sexual spores. And those sexual spores are a little bit different. They are wind dispersed and they are also more resistant against the environmental conditions. Those will contribute to the epidemic in the same year, but they will also act as primary inoculum for the next season's epidemic. So those wind dispersed sexual spores will survive over the period from harvest to emergence of the new crop. And they will start infecting the seedlings. And it's really important for us to be able to understand how the disease works in season scale, and also more precisely in which conditions and how it works. So for example, in terms of weather, what the disease needs, what kind of temperature, what kind of rainfall. Because when we understand what the disease needs, we can try to remove those conditions. So we can try to make conditions if we can affect it, so that the disease is not thriving. And also, if we know that the conditions, environmental conditions, have been really good for the disease, then it makes sense to, for example, spray fungicides to control the disease. If the conditions are not good for the disease, there is no need for control. And this leads to the main target of my thesis work, which is to improve control of Septoria triticum blotch. We had different objectives. First, we wanted to characterize resistance of wheat cultivars against Septoria triticum blotch. Second, we wanted to measure epidemic parameters with high precision. These two objectives first will lead to improved understanding of what we have in hand for controlling the disease. And the second will allow us to understand the disease dynamics more precisely, probably do more precise models of the disease dynamics to predict the disease, and also try to understand it better for helping our management. And in the third step, we would like to improve the control with some kind of clever strategies. So, in the first chapter, we had the objective of characterizing resistance of wheat. So we measured quantitative resistance of wheat cultivars. We had 335 wheat varieties growing in a field, and they were naturally infected by local Gynoseptoria population. We went to the field and collected in total 22,000 leaves, and we sticked them on those paper sheets, scanned them, and got high quality images of the leaves. Then we used automated image analysis to measure disease severity on those leaves. In the image analysis, we took the leaves and analyzed it for different traits. First, we have a kind of a traditional trait of disease severity, which reflects host damage. So what is the proportion of the leaf tissue that is damaged? Some cultivars might have small lesions or small number of lesions. Some of them might have larger or higher number of lesions. In total, we had 37 square meters of leaf surface that we analyzed, and 11 square meters of that was damaged, according to our image analysis. But thanks to the image analysis, we were also able to measure smaller symptoms, those pycnidia, the dark spots. Here we could detect those dark spots within lesions, and we found out that in some cultivars the pycnidia are with low density, they are with low density, and in some cultivars with higher density. And this pycnidia density is important for the pathogen. It reflects the pathogen reproduction, and it also reflects the potential for consequent epidemics. So how many spores the disease the fungus is able to produce for further infections. In total, we counted around 3 million pycnidia on those leaves. So you can imagine that with manual counting it wouldn't be possible, only to this, thanks to this wonderful image analysis. So now we wanted to characterize the wheat cultivars based on these traits. First, host damage. The x-axis reflects the host damage values, and the individual blue spots are for individual leaves. The sigmoidal curve, the S-curve, gives the mean values of each cultivar. So there are gray and colored points for mean host damage of each cultivar. And those cultivars are now sorted according to the host damage levels. So that in the bottom, we have cultivars that have on average low host damage. And higher up, we have those cultivars that have higher average host damage. So it's good to be in the bottom and bad to be on top. And now we did the same thing with pathogen reproduction. Again, we have methods of pathogen reproduction on individual leaves. We have means of each cultivar, and we have ranked the cultivars according to the pathogen reproduction. In the bottom, there are cultivars with really low density of Pycnidia, so those cultivars are allowing for low levels of pathogen reproduction. Higher up, we have cultivars that have really high density of Pycnidia on their lesions, so they are allowing for higher levels of pathogen reproduction, which will then allow for faster or stronger epidemic development. And now, the natural question was, of course, what is the connection between these two rankings? Can we predict levels of pathogen reproduction based on host damage? And it turned out that the connection is really not very strong. When we take 30 most resistant cultivars measured for host damage, so the most resistant cultivars against host damage, and map them to the ranking on pathogen reproduction. We have some cultivars that keep their good ranking, they stay in the bottom, but we have really many cultivars that jump to the top of the ranking on pathogen reproduction. Which means that those cultivars have really low host damage levels, but they have really high density of Pycnidia within the lesions that will occur. And also, other way around, when we took the 30 most susceptible cultivars against host damage, those that allow most damage on the leaves, some of them will be susceptible also against pathogen reproduction, but some of them were really good against pathogen reproduction. So even though there were large lesions, there was really low density of Pycnidia within those lesions. So in the first chapter, we came to the conclusion that there are two types of resistance. One type of resistance is against host damage, and the other type is against pathogen reproduction. So that is kind of the main result or highlight of the first chapter. Now I will also show little highlights from the other chapters. In the second chapter, we move to the second objective of measuring epidemic parameters. And here what we measured was infection efficiency, which means the probability of infection of spores. We took aluminium plate where we put healthy living leaves, and we flattened them on the surface, and then we sprayed those plates with spore suspension in different densities or different concentrations. After spraying, we took those reference membranes between the leaf sets, and we were able to determine the density of spores that we were actually spraying. So there were some variation in the spraying procedure, but because of these references, we were able to measure it afterwards, what actually ended up on the plates. Then we took photos of the leaf sets every day and counted lesions to get the total number of infections. And this work was done, the practical work was mostly done by Susanne and Nora. So when we look at the connection between spore density and lesion number or lesion density, with low spore density, we have practically no disease. With medium low density, we start to see some lesions appearing. With medium high density, even more. And with the highest density, the leaves were totally infected and destroyed quite fast. And from the scope of that connection, so how much spores was required to create certain increase in the lesion numbers, we determined that the infection efficiency was 4% in our case. In the third chapter, we measured another epidemic parameter, spread of the disease in the field. We had elongated plots like this In the middle we had the inoculation area where we sprayed spore suspensions of different strains. Then we went to the field and inspected the disease levels in the inoculation area and also outside of the inoculation area on those light orange measurement lines. We again collected the leaves for measuring the disease severity, but we also used genotyping. We isolated some strains and we used strain-specific PCR markers that we developed for detecting our strain. When we sprayed our strain in the inoculation area, we were able to confirm that it was our strain that was spreading out on the plots, and we were able to differentiate between our strain and the natural background infection from external sources. As a result of the phenotypic characterization, so the disease levels that we observed, first on the left we have differences in the inoculation area. So one strain, the green one, 3D7, was producing more Pygnidia than blue one, 1A5. And then after one spread event, we measured the disease gradients. They were quite beautiful, I would say. You see clearly that close to the source, we have higher levels of disease, and further away from the source, the disease levels go down. And the red line reflects the disease levels on uninoculated control plots. So it's kind of natural background level of infection. So in the furthermost points, the disease levels were down to natural level. From there was no contribution from our source anymore. We genotyped some strains and also got the same pattern. At the closest measurement point, we had around 80% of the isolated strains being our own, and only 20% from external sources. But further away, only 30% of the infection was from our strains, and most of the disease was coming from external sources. And now, when we fitted the model to the phenotypic data, we were able to characterize the spread and come to the conclusion that when spreading from a single point source, the disease will create certain pattern, and 99% of the new infection that is caused by the spread from that source are staying within 1 meter distance from the source. And finally, in the fourth chapter, we started to implement some clever strategies for controlling SDB. And here the clever strategy was cultivar mixtures. We had 3 cultivars: susceptible Karim, the yellow one on top, and then 2 resistant cultivars, Salim and Monastir, blue and green. The experimental design was made so that we always had Karim in 0, 25, 50, 75 or 100%. And then we complemented the Karim with either Salim or Monastir or their combination, their mixture. So we put a certain percentage of the resistant cultivars. So for example, here we have 25% of Karim and 75% of Monastir, and then 75% of Karim and 25% of Salim. Probably not for a surprise anymore, we went to the field, collected the leaves, and used the automated image analysis for measuring the disease levels. And when we look at the disease level data from those mixture plots, we were really happy to see quite clear results. Here in the plot, all of the treatments are pooled based on the percentage of resistant cultivar. On left, we have 0% of resistance, which means the susceptible cultivar alone. Then we have 25% of resistant cultivar added to the susceptible, 50%, 75%, and 100%. So at the end, we had only the resistant cultivar alone. And when you look at the disease levels that we measured, you see clearly that the susceptible cultivar alone had higher disease levels, and all of the mixture treatments had lower disease levels, and those were not significantly different from each other. And this gave us a really nice conclusion that already 25% of resistance in the mixture is enough for suppressing the disease levels as much as the resistant cultivar alone. We don't need to show entire field with our resistant cultivar. It's enough to put only 1/4 of the seeds being resistant, and the rest can be susceptible. So, to summarize these highlights from my chapters, we observed these 2 types of resistance against host damage and pathogen reproduction. We measured 4% infection efficiency and around 1 meter spread of the disease in field conditions. And finally, we found out that 25% of resistance is enough for suppressing the disease very efficiently. As a conclusion, I would say that epidemiology is really useful. Of course, I love it. And I think that there is great potential for further improvement of plant disease management. Thank you very much.
Thank you very much, Petteri, for this nice presentation. Even as an atmospheric scientist, I know something about diseases. I suggest we go with Emerson Del Ponte as an independent examiner first. You can take whatever 10-15 minutes to ask questions, and then we go for a further round if required.
Okay, thank you very much. I'd like to first thank you, Petteri, for the invitation and also the committee, Professor Bruce and Professor Alexei. It's really a pleasure to be here, coming from a long and far away country. It's a long trip, and I'm fine, I'm feeling well. Yes, so you are here you're showing your work that I've done during 3 years, right? This is 3 years of
3
work.
years, yes.
3 years, and you have 4 chapters.
Mm-hmm.
So you were able to put out 4 chapters within 3 years, which is really great. It's really, when I was reading your work, I could see a lot of work. So you worked very hard. But obviously, this is not an independent work. You have help from the team.
Yes.
This is really nice that you have the teamwork. So many people helping you, which is good. And you had a mix of work from the DNA up to the field. You could learn a lot of different concepts, different tools and approaches, learning from the lab, from the bench, going to the greenhouse and potted plants and going into the field.
Yeah.
So this gave you a really nice overview and broad techniques, and you learned a lot of concepts about epidemiology. So this is really a very classic epidemiology work, which is really good. I like it very much. So. But within 3 years, and you've been so busy doing work, I wonder how many epidemiology books you've read during this time. Because I know that you don't have classes here, so you did not attend any epidemiology course formally, but you got access to textbooks, to articles, and to the literature, which is important for you to base your planning and research, and all your work is based upon what has been done before you, right?
Yes.
And I have some questions about some concepts. Okay, when I was reading as an epidemiologist myself, I like to check if you know well some of these concepts, which is really important for you as a doctor in epidemiology. So everybody will look at your thesis and say, wow, this guy is an epidemiologist, because the kind of work you did allows you to say, well, I did a lot of base epidemiology work, and not only experimental, but you did a little bit of modeling too. Okay. When you mentioned about the host damage, is there any other term in the literature that refers to host damage that's more intuitive and more, I think it's a better term?
Severity?
Yes, but when you mean host damage, is that damage can be visible damage, invisible damage. Damage is quite broad, it doesn't say much. Is there any other term that you could use instead of host damage and pathogen reproduction? Because you assessed something that was visible.
Yes.
Right? Not only for our eyes, but for the camera.
Yes.
So you could pick colors and stuff. And is there any other term?
Besides severity, I don't think I'm aware of any term for that would be somehow similar.
Have you heard of symptoms? Symptoms.
Symptoms, yes.
Yeah, that's what we see.
Ah, yeah.
The
So
host
sure.
damage, I think what you measured, what you quantified was the symptoms.
Yeah, sure.
Yeah, symptoms.
Yeah, true.
And in pharmacology, we say that when we see something on top of those symptoms, we call signs, which is the reproduction part of the pathogen. And it's more intuitive. And there's another term that you have used to refer to this amount of these symptoms on the leaf. You call it PLACL. So this is an acronym.
Yeah,
that you
but
created.
so
This is based on the previous paper, right?
yeah.
So, but there is another term that explains that acronym, right? And you just mentioned that.
You mean severity?
Yes.
Yes.
Yeah, that's more. It can be a kind of vague term because people use for different things like the index, and they use these across different contexts. And I think you define it well, but that's very intuitive, and we have a proportionate effect of the disease.
Yes.
By the symptoms that are visible.
So when we were writing the first paper, we were thinking of this terminology, and of course we realized that the severity is a much more common word for the host damage, for the PLACL. But then sometimes severity is also defined based on percentage of leaf area covered by Pygnidia bearing lesions or something like that, based on the Pygnidia. And we thought that we could refer to host damage and also the pathogen reproduction with severity. And we wanted to make clear distinction so we don't use this perhaps more intuitive but probably less precisely defined terminology.
Yeah, I see that you try to find a more specialized term to facilitate the communication between people working on this disease, to make clear that you were referring to symptoms and signs and things like that. And when you mention your title is quite broad, it's measuring and controlling, right? Measuring and controlling the pathogen, right? And when we mention epidemiology, and is it epidemiology of the disease or the pathogen? You control the disease or you control the pathogen?
I would say we control the pathogen.
How you control the pathogen?
Well, of course, it's based on the disease symptoms, and we want to suppress the disease. But well, I always think of it as a kind of management of the pathogen population. We want to suppress or somehow manage the pathogen population. Sometimes we probably don't want to suppress too much, or not creating so much selection pressure for the pathogen.
But you measure to make sure that you control the pathogen as well. You measure the disease.
Yeah, we measure the disease. Yes,
the disease.
That's right.
And all the processes, they depend on the host too. So if you use a cultivar that suppresses the pathogen, you're not actually killing the pathogen like using a fungicide. Then you control the pathogen.
Yeah, that's true.
Yeah.
That's true.
But I think the most correct you would say that epidemiology refers to the disease
of the
processes, and then you can control the pathogen. But I think it's more in our field we say we control the disease. We can suppress disease, or depending on the kind of a pathogen life cycle, you can apply something that's is targeting the pathogen or the plant.
Yes.
Usually our target is the plant. You protect the plant.
Yes.
You try to modify the plant.
Yeah. And of course the eventual goal is to reduce the disease.
When you say measuring, there is one section in your text. You say you measure the quantitative resistance. Actually, you measure.
We measure
the quantitative resistance, or
we measure severity of the disease.
you
and
quantify.
Yes, we quantify the disease levels, and so then we use that as a proxy of the quantitative resistance. So if we see lower severity of the disease, then we assume that it's
okay. You are measuring the traits that are associated with the resistance.
Yeah.
Just to make sure these terms are okay or not.
Yeah, it was
you
also
gotta read carefully. When you read carefully your work, I could take a lot of these inconsistencies with the previous literature.
Yeah, we had discussion with the reviewers about the use of component of resistance. For example, you want that to be really careful for not to refer too directly to the resistance when we are actually measuring the disease.
And actually, you are at the edge of a field that there is a barrier. We have a plant pathologist in one side, we have the breeders in the other side. So there is a jargon that is specific to each of these groups. So then, eventually, there's a collision. You don't agree, and you need to make sure you use something that is understandable by both groups. Okay. But it's still, you have a literature to give you feedback or background on this. Okay, I'd like to talk a little bit about chapter 1. And you had this high number of cultivars, more than 300, right? They were planted at the same time.
Yes.
So are they synchronous in terms of phenology?
No, they are not perfectly synchronous. And we have some data about flowering, but we didn't include that in the analysis of the.
And they probably have different shapes and heights.
Yes, they have different shapes and heights.
Size of the leaves.
Leaf size and distance between leaves and they are of course timing.
And do you this is a factor? This could be a factor affecting the results?
Yeah, sure. For example, if we have some higher cultivars that manage to escape the disease, the leaves grow, trees emerge and the stem elongate so much that the healthy leaves are kind of away from the disease source.
But you could not account for these factors.
No,
because you were just sampling leaves within the same plot at the same time. You sampled over 2 times.
Yes.
It was before flowering and after flowering.
Yes.
Right? About 1 month.
1 month, a little bit more, 5 weeks.
So we did not have more precise assessments over time, just visually. Visually, you had 3 assessments. Visually, and then using the image analysis, you sampled over 2 times.
Yes, 2 times.
And you sampled a few leaves. How many leaves you obtained from each plot? About 45 leaves? From
each plot, at each time point, we got about 16 leaves. We had 2 replicates.
16?
Yes.
And then you had another?
Yeah, we had 2 complete replicates, so we got around 32 leaves for each time point, and then 2 time points, about 60 leaves in total.
These replicates you planted at the same time?
Yes, also? Yes. They were something like 100 meters
block.
Yes.
Yes. Okay, what is the trade-off of sampling fewer leaves, let's say, fewer leaves, and get a great amount of data from fewer leaves instead of sampling more leaves and get a more rough measurement of the disease in your case?
Yeah, clearly there is strong trade-off. If we collect really really large samples, then we can't characterize them so precisely. And for example, one of the weaknesses of the work in the first chapter was that we were not able to characterize the disease levels, the overall disease levels in the plots, because we were collecting infected leaves. Okay. So we don't know exactly which proportion of the leaves were infected. We don't know the incidence of the disease in those plots.
Yes, so you could not really track the epidemics over time using this method. You just you sampled during 2 different times, and you could see differences in terms of the disease levels. So first time you have less disease, and then you have more.
Yes, yeah, and those were also
and
different
you could predict
so
the second time based on the level that you could see the first time, and then you could use that information to predict. Okay, but all the epidemics they were in the same area, in the same environment.
Yes.
I was a little bit curious. I have a question first about this new index. You have this PLACL, and you have the number of picnidia per lesion, right?
Yes.
It's per size of lesion or per
Yes,
lesion?
per unit area of
Per
lesion.
unit area of lesion area.
Lesion areas, yes.
And the index was confused about how you calculate it, because in the manuscript it's not clear to me how you calculate. I tried to calculate using some of those numbers. So it's the number of picnidia per leaf, the total leaf, or you multiply something like. I
You mean how we calculate the picnidia density? So that's
the picnidia density for the leaf.
Yes. So we have total number of picnidia for the leaf, and we have total amount of damaged leaf tissue or diseased leaf area, and then density comes from that, dividing the total number of picnidia on that leaf by
the total area with the symptoms.
Yeah, total area of symptoms.
Yeah.
Then
So
we-
it's not. So what's the difference when you first you calculate the number of picnidia per unit area of the lesion?
Well, that's the final measure. But what the macro actually does is it just measures the symptomatic area and the total leaf area, and it counts picnidia. And then this picnidia density we got by dividing the total number of picnidia on the leaf by the symptomatic area on the.
So this gives you the raw leaf that you have, whole leaf.
Raw lesion. If we divide the total picnidia number
yes.
with symptomatic area, then it's picnidia density in lesions. And then if we divide it by the total leaf area, then it's the picnidia density.
Okay, it's a kind of index. You multiply the first measure by the other measure, right? It's a kind of index. Yeah, it was a little bit confusing to me because you used this acronym, the PLOC, and then you have the raw leaf, and the raw leaf was a little bit complicated to understand how you got to those measurements. And you could have just simply used the percent area affected by the lesions and the number of picnidia per lesion and the index, picnidia index. Because you actually have an index. When you multiply two different things, you have an index. But when you assess it using both of these measures, let's say these variables that you could not differentiate much the cultivar. There was a large block of cultivars.
Yes.
You could not.
Most of the cultivars were
And
in
you
the
have some
medium
on the top
range.
and some at the bottom.
Yeah, so we had most of the differences in the end of the distribution. And actually here the colored different colors reflect the statistical differences between cultivars that we were able to find. So all these orange dots here reflect cultivars that are not different from each other, but they are different from the differential color, so the two extremes.
And do you know why you could not tell them apart? Do you know why? That's all the background that you have, all those points are it's the variation that you have.
Yeah, it's individual.
Throughout the leaves.
Yes.
You have a lot of variation.
Yes.
Even using this very precise, maybe because you use a very precise method, you could make this variation that maybe using the visual symptoms you could not get. So this variation is so large. And so you can see that for some cultivars in the middle, you can have a very low number of media, high number, or the area factors can be very different.
Yeah, it's really variable within each cultivar. And I think this shape really reflects the traits of the cultivars that we had. So most of the cultivars are kind of average or performing moderately well. Only some of them are exceptionally good, so that we can tell them apart from the rest, and also some are exceptionally bad. And actually, when we did similar plots or rankings with the visual score, we were able to we were even less able to tell them apart, and that's likely because those fields were treated by fungicides. So even though it was really bad year for the farmers, so it was really high levels of septoria on fields generally, in our study, the levels were kind of moderate, and probably that affects our ability to really tell them apart. Probably without fungicides, we would get larger differences or more pronounced differences. But then we would most likely have also other diseases that might affect.
I wonder if you got any information, previous information about those cultivars according to resistance level, and then you could check if using this method they would group the same. They would group the same as the previous. I don't know. Do you have this information about the cultivars?
Yeah. So. Some of these cultivars were used in smaller field experiment in previous year, and there was significant correlation between the rankings, but it was kind of moderate or moderately weak correlation. And when I spoke with some breeders, they thought that our correlations are actually really high in their view, and they say that it's most likely because it's so difficult to tell the cultivars apart. So if we try to get correlation between two rankings, most of the cultivars are in the middle range. So it's really easy to get just by random large difference in the ranking.
You have only very few groups, like 3 or 4.
Yes. Yes.
When you use the visual assessment, you split more into groups, or
no? In the visual scoring, we were not even able to split. So I can show you one of the figures in the. So here are the AUDPC values from plots, and these individual dots reflect the basically contribution of each visual assessment to the AUDPC. So they reflect the disease levels on those plots. And as we see here, all of the most of the cultivars has really similar scoring for this time point, and also here there were not so many differences. And on 1 to 9 scale on visual scoring, we had I think highest score was 4 overall. So there were not really high levels of disease.
Okay, one question more about this chapter 1. Did you recommend this method, the image analysis tool? Is it ready to be used by breeders, by the plant-pathologists, or is there something that you just use this method to learn more about what to look for in the future? So visual assessments can be improved. Did you think about this? Because when I was reading, it was not clear to me what the take-home method was. If you're really proposing that people use this it's very complicated. You take all the photos and you present the images. There is the macro, but still it's a lot of work.
Yes.
To get to the image. Yeah. So what is there's something I was just wondering if the visual assessments could be improved just because you know better what to look for now. If you use a method that is very precise. So did you think of any standard area diagrams that you could build just using this database of images that maybe people could use to rate score faster?
Yeah, that might be a really good idea for individual lead scoring, and in our measurements, the measurements based on good media numbers were more robust against the temporal or spatial variation. So I think it would really make sense to try to account for good media in the visual scoring. But then, of course, it's more subjective nature of accounting for the good media density. And I think here there is kind of similar trade-off, as you said before, between high intensity and high precision of measurements, but smaller sample size, and then large sample size with lower intensity of measurements. So with the image analysis, it really requires some work, and I think really well-trained plant pathologists can probably deliver as reliable results with visual scoring much faster. But I think the main audience for this or target group for this method would be academic plant pathologists. All the breeders that I have spoken with say that it's not possible. They have millions of breeding plants. They are not going to collect individually from all of the plots. That's for the breed and would be 1 and 3 like this. So but all the method is open source. It's free, and source code is available, and we are trying to develop it further. And I know that several plant pathology groups are using it in Europe and I think also in Australia.
This screening work is done out in the field, or they use product plants in the greenhouse to screen for this pathogen, for this screening genotypes is done in the field?
In the breeding.
Yeah, breeding.
In the field.
In the field.
Yes.
Yeah. I was just wondering how maybe you can have some good information about, maybe you can improve this image analysis too. Now that you know where to look at,
yes.
Maybe you can like have something like taking photos, or like small photos from
of the plots.
Higher number of leaves across different heights, and then you have this precise area, and then you take a lot of images, and then you process those images instead of taking an image from the whole leaf.
Yeah.
You just go to the field, use your phone, and you can take very precise measurements.
Yeah, we know there are a couple of companies that are developing mobile device applications for at least detecting some diseases, and we also tried that, and we also took some steps towards the indirect plot measurement that you mentioned. So we had photos of the canopy, and we tried to see if we can visually detect lesions, and we think that if we can count lesions or somehow detect them visually, then probably machine can do it also. But we were not able to take that really far. But in one more promising line, we collaborate with Chrome Science people, and they used our leaf level data and their hyperspectral measurement data. So they had a camera of the field, and they took hyperspectral images. And there are some promises for faster automated pixel measurement.
Just one final one, just missed. You did not connect.
Then we should move on.
Yeah.
Now you're at good time. So this is your last one.
The last one, okay. You did not have new data from this file. Did you get new data?
No.
Wouldn't that be important to have new
Yes.
data? Because you are trying to say what kind of variables is most important, but really for a farmer, he would like to have a good crop.
Yes.
That is still that
Yield matters, yes. So here we need to rely on some previous work that show that disease levels on black leaf and black mouse one leaves are important for the yield production. But the perfect case here would be to have a complete box of the cultivars growing in disease-free conditions to be able to differentiate between this yield without the disease and yield with the disease.
Okay, I'm fine now. I'm done. Thank you very much.
We can always come back.
Yeah,
Sure.
sure.
But we should move. Give Alexi a turn here. He doesn't get any rest, but you can keep it going here.
So thank you very much, Petteri, for your talk, and I have to say that I feel very lucky to be able to work with you during the last 3 years and to advise your PhD research. Great job, very well done. So I have several questions about kind of focusing on a bigger picture of the population biology of this pathogen, in particular from the evolutionary perspective, because I know that you'll be working more with evolution in the nearest future. So experts on this pathogen often say that the population is very diverse genetically. So the first question is, do you think this genetic diversity also translates translates into high phenotypic diversity of different strains?
I think so. Yes. So when we measure any phenotype of the pathogen, either in planta or in vitro, we always see lots of differences between strains, be it fungicide resistance or latent period or lesion size or pupillary production on different cultivars. And then of course it depends on the conditions.
Right. Then to follow up on that, this is a pathogen of an important crop, wheat, and people are trying to control the disease. And in this way, they are also suppressing the pathogen population. So we think that the population is under strong directional selection. And kind of what evolutionary theory tells us that if a population is under strong directional selection, the diversity should decrease over time. But that's not really what we see. How is this population able to maintain this extremely high levels of genetic diversity and corresponding phenotypic diversity?
Yeah, so I think the case here is that we are not really able to suppress the pathogen population significantly. So there are always fields where the pathogen can survive very well, and even in best fields of resistant crop treated with fungicide, we have some levels of disease. And there are also, for example, in fungicide spraying, not all the leaves are perfectly covered by the fungicide. And in the bottom of the canopy, on dead leaves, the host resistance is not active anymore, but the pathogen can survive there. And then when it's having sexual reproduction every year, it's mixing basically all the material that's around. And if I remember correctly, one estimate was that 90% of global genetic variation is present in just a single field when we sample intensively and measure the diversity. So we can't suppress the diversity.
So okay, you said that we cannot suppress sufficiently the pathogen population, but how does this inability to suppress it, how does it allow the maintenance of the diversity?
Well, there's if you think about the directional selection, I think what I mean with not suppressing the pathogen is that we are not really imposing the directional selection on entire population. Only part of the population is affected by the selection, and large part of the population is not under the selection, and that's one way of maintaining.
But okay, you can have a large part of the population, but why should it become diverse? I mean, what are the mechanisms that
because of sexual reproduction, the genetic material from different strains from the part of the population that is under the strong selection and from the part of the population that is not under the selection, they mix, and then for example, if we are selecting for really fungicide resistant strains on fungicide treated leaves, we might get some reduction of diversity there or strong directional selection. But any strain can actually land on that lesion and mate with the strain, which means that the strain that was not under selection can mate with the selected strain and kind of break the genetic combinations. We can't continuously keep same strains under selection, except in greenhouse.
Okay. So then a bit more specific questions on the chapters. So related to what Emerson was asking. So what we see, if you could show the previous figure. So what we see is a very high, like the outcomes within each cultivars. There's a very high variation.
Yes.
As Emerson pointed out. So what do you think are the sources, the main sources of this variation? And on top of that, how, if you would be able to follow up on this experiment, what would be a way to disentangle and tease apart these different sources of variation? within each cultivar.
Within each cultivar, yes. So I think there are some stochasticity, of course, where the spores land and which leaves happen to be in favorable microclimate or things like that. Then in the fungicide spraying there can be heterogeneity. Some leaves might get more fungicides and some leaves don't. And what else I have in mind? Well, something that we observed in the second chapter was that when we inoculate individual cultivar with individual strain, so genetic material is the same. We keep them in same environment in the greenhouse. We see some lesions appearing after 2 weeks, and some lesions appearing only after 5 weeks. So there's lots of unknown variation or variation that's not known where it comes. When all the genetics and environment is the same, there's a lot of difference in the development of disease. And how to account for different sources? Well, I think with the fungicide, it might be possible to have fungicide treated and non-treated plots to see if that affects it. And then probably when we were sampling, we should define better. Probably train the people collecting the leaves better, so that they are not by accident collecting naturally senescent leaves or leaves on the borders of the plot and things like that, to try to keep the sample quality as good as possible.
But so, I mean, related to my previous question about the diversity. So, and this is a natural infection. So what?
So one part of the variation is as the stochasticity in the spore landing where the inoculum is present.
Yes.
But also when the inoculum is present, which genotypes, which strains are present there. Some strains might be much more aggressive. If we get just a single lesion of aggressive strain in leaf, it creates a Wikipedia, and then those spores will spread over that leaf really easily, and then that particular leaf might be much more diseased than others. And then of course we can account for that by measuring the genetic diversity or observing some isolates on the leaves.
Yeah. Okay. Thank you. So. Now about the chapter 2 on the measurement of infection efficiency. So the measurements you conducted were kind of a proof of principle, and so you looked at infection efficiency and the latent periods in one particular pathogen strain and in one which genotype.
Yes.
So do you expect? I mean, partly you already answered that. So I think you would expect a lot of variation between different strains in these parameters. Yes. But then the question is, how many strains would you need to characterize in order to really kind of capture the characteristic distribution to characterize the whole population?
That's a good question. How many strains? I think that that might be. We might approach that with similar methods as with the study that I mentioned, where they find that 90% of global variation, genetic variation, is present in one field. So we might, if we had really large isolated collection, and then we know something about the global genetic variation, we might kind of try different sample sizes to see where we achieve some level of 80% or 90% or something like that of the global variation.
Mm-hmm.
Then of course we don't know if that genetic variation that we observe is really related to our phenotype, but I think that would be my guess.
Mm-hmm. So let's move on to the next chapter about the dispersal. So do you think this parameter of dispersal, the dispersal radius, is it a trait that's under selection in this population? And if so, what kind of selection?
It's hard to see how the pathogen would really affect the spread of the spores. I mean, there is some variation in the spore size, and but the spores are anyway really small compared to the size of droplets of rain splash. So I think that that might not affect it. But one thing that pathogen can do is to make sure that the spores are available when spread is possible. So when rain event, when we have rain, and one way for that might be what we see in the heterogeneity of latent periods. Some regions after 2 weeks they are ready, waiting for rain from 2 weeks to probably 3 weeks. I don't know when the spore will die, but then some fresh spores will appear later in 4 or 5 weeks.
Okay, you could get. Maybe you're one more question.
Okay. So now, one question about the last chapter on cultural mixtures. So, if you are able to repeat this experiment and have a large team doing any kind of measurements that you like, so what would be a way to distinguish the contributions of different mechanisms of disease suppression in cultural mixtures? For example, distinguishing between the disease suppression due to the density effect or dilution effect and the barrier effect. I mean, how would you go about doing that?
Well, if I had infinite resources, then it could be possible to plant poor stands of the cultivars in different densities to see how the density affects it. Then we will probably put some physical barriers, not plants, but or probably some other plants, but even better kind of artificial wheat plants. Mix them with the canopy.
Or non-host, maybe
probably non-host, like barley or something like that. And then one thing that would be really interesting would be what's the level of epidemic on different components of the mixture. And the French people got really nice work on that. I think it's not published yet, but they colored the seeds before sowing, so that when they go to the field to pick the seedlings, they were still able to see the seed and see if it's resistant also to cultivar. And then later on, the canopy structure was more or less similar, except that one cultivar was with awns and the one was without. So after heading, they were again able to differentiate between resistant and susceptible cultivar.
Okay, thank you.
Okay. I'll try to keep it to under 20 minutes. So first of all, I just want to say I really liked your general conclusions chapter. I think it was very well put together. This is where you summarized all of your findings and sort of put them into an application sort of network. How would you apply these findings? And I think I'm tempted to make this mandatory reading for all of our PhD students, because I think you did such a nice summary of how this research translates into application. That was very nice. Thank you for that. And I want to start with a question on your fourth chapter, which was the mixture experiment. And I don't know if you're aware of the fact, when I was a PhD student more than 30 years ago, my PhD research was on mixture effects, and that was with barley plants and barley scald pathogen. And what I found was that once you got to 33% resistance percentage in the mixture, you got a significant mixture effect. You got 25%. And the reason I say this is these mixture experiments go back at least 50 years, and they're deep and broad around the world, and they 95% of the time show a really profound positive influence on diseases, and a positive influence on yield, and a positive influence on yield stability.
Nutrient use, water use,
everything.
Yes.
So why are we still growing clonal monocultures? What the scientific data is so strong and exhaustive. Why are we still doing clonal monocultures?
Yeah, I think there's bad gap. First of all, there's big gap from academic research to applications. I think it's quite common that when academic scientists looks at some problem like this is that we are looking at here, they see the problem, they study it, they find a solution, then they publish the solution and they think the problem is solved. It's all only in their mind, but no one is using the solution. And when I presented the cultivar mixture work in International Symposium of Cereal leaf blights a few weeks ago, after the talk, one breeder said I was asked about her opinion on the cultivar mixture, and her opinion was it's kind of a nice idea, but it will never work. From her point of view.
Did she say why?
Well, I think her idea was that it will never work for the breeders, and I don't think even that is true. But with their current system of how they plant the millions of breeding lines, how they score for the traits and things like that, it would require really major changes in all their approaches. And then she was speaking about introducing mixtures. The mixture breeding aside of the conventional breeding, they would require double resources for everything and like that. But probably some smaller breeding company, like a young startup, might take up some ideas and use them. But I really see more potential in trying to convince farmers to use mixtures, because they can use mixtures. They can buy seeds and mix them themselves, and in quite many markets it's possible to sell mixed seed.
So if you get the farmers doing their own experiments and seeing positive outcomes, then they're more likely to adopt that technology.
Yes.
So really, we shouldn't be communicating to the breeding companies, we should be communicating directly to the farmers.
Yeah.
And once you figure out how to do that, please tell me. Okay, so now I'll go to chapter 2, and this wonderful number you came up with, which is 4%. So about 4% of the spores applied onto the leaves on average formed a lesion. Now that number is an interesting number. Do you know how well that number compares with similar experiments done with other pathogens?
With other pathogens, there was review on infection efficiency measurements. The methods are really variable, so the methodology is not directly comparable. In some cases, they get over 50% efficiencies, and in some cases, it's around 1 to 10%. So I think it fits well, but it's probably in the lower end of the spectrum. And in one experiment of Helen Foes and others, they also got as kind of side result. They got infection efficiency, and in their case, it was around 50%.
Wow. There's a big difference. Same organisms, greenhouse experiments in both cases, 4% versus 50%. Which result do you trust more?
Well, both of the results are with single cultivar, single strain, and in single conditions. So I wouldn't make really strong conclusions based on either of those, especially when we have two really different results. Even guessing in the middle would be bad.
Yeah.
So yeah, I would like to measure it a couple of times more.
So in science, when you get these types of dichotomous results, different research groups working with the same organism, asking the same question, maybe using different approaches, but they come to wildly different numbers, what's a good strategy for finding the truth? How would you propose to find the truth there?
So I think first of all we would like to look at the possible reasons of the different results. So starting from the strain and cultivar that we are using, we could measure those genetically identical combinations in different conditions. Then inoculation method might affect. In their case, they put droplet on leaf and then spread with finger, and we sprayed fog of the spore suspension, and that might affect the placement of the spores, their ability to penetrate, things like that. And then, of course, the greenhouse conditions. So first, I would like to try to unify the methods and conditions, and then try similar genetic material.
Surprisingly enough, I think that your findings are more likely to be correct, closer to the truth than their findings when they rub the spores into the leaves with a finger, for example, introducing all kinds of wounds. But finally, I think it's important that people understand the problems with all these methods and again, try to design something together. To get all the advantages of different approaches. Okay, one more question. And that's with the dispersal kernel experiment that you did. 99% of new infections fall within 1 meter of the source. How does that result compare with other experiments done by other people in other pathogens?
It compares very well. The mean dispersal is in the range of previous measurements starting from, I think, 70s already, where they measured dispersal of septorial nodorum or septorial 3DC. The range class dispersed pretty similar pathogens. The mean dispersal distance with range class is usually around 20 centimeters or something like that. So it's pretty similar.
Okay. So it's really believable.
Yeah.
Okay, I think that's enough. Take it to.
Thank you. We're well past the hour, but if there's 1 or 2 more questions you would like to ask, then.
I have one.
Okay.
Think about this question, the previous one about the number of lesions is not so high in this disease, right? You could not see, so that the infection efficiency or infection rate, when you compare the number of viable spores and the number of lesions, you really have few number of lesions. You could not see, you have more these expanding lesions that are larger lesions instead of having a high number of small lesions. And you also notice that the latency period is quite long.
Yes.
Okay. You could not really, if you thought about the fact that maybe you have infections, you have successful infections, but it takes a lot of time, it takes a long time. So the lesions that appear start to grow faster, they go over the other lesions. Would there be a way that you could use another technique that you could really mark the initial infection of these lesions? Because when you use your method, it was vision. You were just counting the new lesions over time.
Yes.
But that doesn't mean that you could not see the invisible ones. It takes a long time.
Yes.
So maybe there is a real, let's say, actual infection efficient that you cannot see, and that it's because this pathogen tends to the kind of damage that it does on the leaves just grows quickly and overshadow the other lesions. So just a thing I was having other activities. Maybe it explains because it's something that the disease is formed by lesions that are a little bit larger.
Yes.
And you provided data that about the latency period is very variable across the different lesions.
Yes.
Is there any technique that you?
So what we did was that we took photos every day on the leaves, because we often notice that we see young lesions, just the first yellow chlorotic spot in the leaf, and then another spot appearing probably 1 centimeter away from that, and 2 days later they would be merged. So by counting the lesion number every day, the newly appearing lesions, I believe we were able to get more precise number of total number of infections. But in the data we also noticed that there was saturation effect. So when we used different densities, we got this linear relationship with low density. So probably there the lesions were not overlapping too much. But with highest density, we got clear saturation, because the first set of lesions in 2 weeks was enough to cover the leaf, and then of course we don't see if anything is successful afterwards.
Yeah, but my question is there any method that we could track the invisible infection sites just before, because we track the visible symptoms?
Yeah,
so my idea is that there's something, there is a lot of infection sites that you cannot see. But when you see the symptom, it's overshadowing all the other infection sites that you could not account for.
There is actually some work done on the epiphytic growth of the spores or the hyphae on leaf surface, and they measured the penetration success based on microscopy. So they were able to see when the spores actually penetrate, and I think in their case the penetration success was again higher than for us. So it might be that some of the visible penetrations, microscopically visible penetrations, do not lead to successful infection. And they also observed that some penetrations happened after 1 day, and some happened as late as 10 days after inoculation, and that's one factor affecting the variability in latent period. And then, of course, it's a question whether it's a true latent period that we see, whether we should start counting the latent period from inoculation or just from the penetration, how long it grows inside the leaf, and so on.
Do you have an idea? What would you
do?
Let's discuss that.
Yeah, maybe it's kind of a thermal imaging, some kind of different technique that's more precise that you can maybe track those infection sites, or maybe mark GFT something.
I was thinking GFT for clearing.
I don't know. It was just I don't have a solution in mind, but I think that maybe within hours that we could just track. But that would be the real efficiency. What we have here is the new, not infected, the new lesions over time.
Yeah, and I think it would be interesting. In this group, people developed a method for inoculating leaves with different fluorescent marker strains. So basically same strain with different markers. And then when spraying on the leaf, they get the different colored pygmedia. The spores coming out from pygmedia are of different color. And that might be used for detecting the mixed lesions. If we see that the individual lesion is bearing multiple colors, then it means that the single lesion is coming from several infection events.
The children are trying to rescue Dad. It was very strong signals.
He's getting outside help. I'm not sure.
I've got lots of outside help, I have to say.
Any other questions? Urgent questions from the committee? Questions from the audience? Maybe one or so? Everybody happy? Yes?
Yes, please. You had the dispersal kernels experiment, and the answer was like 1 meter.
Yes.
But then you also mentioned that 90% of these strains are in any field anywhere in the world, which suggests that they're actually really quite good at going global.
Yes.
Is there a discrepancy? And what's the solution?
Well, what we measured was race class dispersal of asexual spores, but the sexual spores are wind dispersed, and they can fly over kilometers or hundreds of kilometers, and that leads to mixing of the genetic material.
So if that's the case, then does it really help that we have this one stage of very short dispersal? I mean, you're kind of like implying that the short dispersal can be used as a control mechanism by manipulating it.
Yes, the latent period for asexual reproduction is much shorter than for sexual. So there can be up to 6 cycles of asexual reproduction and 1 or probably 2 cycles of sexual reproduction in the season. And if we can suppress the asexual part, then we will stop most of the time.
Last one.
There's another question over there. Would you give time?
In your field experiment in Tunisia, you did try to have the 2 resistant cultivars together in association. I don't think you reported on whether it did anything interesting.
I didn't show it, and it didn't do anything interesting.
Thank you.
Okay. Thank you very much for this interesting discussion. We will close the official public part of the defense now, and the committee should stay. We need to discuss for a few minutes, and the rest of you, thank you very much for coming. And don't go too far. We'll be back in a few minutes. Thank you very much. Thanks.
Sorry, these chairs are so confused.
The chair has to go.
Yeah, sitting down is like an IQ test.
We have several days after the interesting discussion. Look forward. You on the oral technology with already in our hands that you had some other ideas for how to count these infections that we have to do.